Very good morning to all the BIPC students. So here we are in our session where we are going to deal with complete short answer type of question from genetics. And genetics, as we know, this is a very significant chapter, not just for IPE, for MSET and NEET this carries huge weightage. So how you can parallelly prepare the concept of genetics both for your IPE and your MSET and NEET? Here we go. First question is about explain the law of dominance using monohybrid cross. Okay, law of dominance using monohybrid cross from Mendelian's laws of inheritance, that is law of dominance, law of segregation, law of independent assortment. These three laws, statement based questions have been asked multiple times in your also. Okay, and same concept is there for your IPE as well. Here how you have to deliver the concept, what you have to learn, I will be explaining you now. Listen, first when it comes to law of dominance, what is law of dominance actually you have to explain. Law of dominance states that from two alleles contrasting pair of alleles come together, one of them will express it and the other remains hidden or masked. So one allele will mask the effect of the other allele. The allele which is masking the effect, allele which is expressing is addressed as dominant allele, and the allele which is masked is addressed as what? Recessive allele. This is recessive allele and this is dominant allele. Okay. The expressed allele is called as dominant allele and the masked one is addressed as recessive allele. So Gregor John Mendel demonstrated this law through what? Monohybrid cross. Okay, he has demonstrated this using monohybrid cross using three plants, that is Pisum sativum, differing in one pair of contrasting traits. For example, this is for tallness and these are the dwarf plants which are homozygous dominant and homozygous recessive ones. Here we go with first parental generation followed by F1 generation and then F2 generation. Parental generation, which is pure tall and pure dwarf. Okay, with two alleles, and in F1 generation this is the hybrid. Okay, and the gametes which are formed over here, capital T small t, capital T small t. Find right. These are four gametes for what? F2 generation. In F2 generation you will get classic. You can also represent this with a checkerboard.
Okay, an example, a person diagnosed with stomach cancer, by using radiotherapy or chemotherapy, we have removed that particular tumor from the stomach.
So the phenotype to 6 months ratio both will be one and the same. Is to capital T small t is to small t, which is 1 is to 2 is to 1. This is both for phenotypic and genotypic ratio. Clear till here? Understood? So explain about the law of dominance followed by the law of dominant allele and recessive allele, and then give out the parental generation, F1 generation, F2 generation with gametes in a clear way. And then monohybrid cross. Here in monohybrid cross you see first parental generation, dominant and recessive you see, and gametes here we have capital T small t. First generation, that is filial generation. Tall plants are tall, only the dominant trait, that is tallness, will be appeared over here and dwarfness will be masked. Okay, that law of dominance on selfing F1 plants, when F1 plant undergoes self pollination. Okay, capital T, capital T small t. Fine, right? This will be seen when all plants are tall, where you will get both phenotypic and genotypic ratio. Phenotypic ratio will be 3:1, where 3 dominant phenotypes and 1 recessive phenotype is seen, whereas genotypic ratio will be 1:2:1. Fine, right? Give out both phenotypic ratio and genotypic ratio in a proper way. Okay, these will be the results of the F2 generation. This experiment will prove that crossing between parental generation, F1 generation followed by F2 generation phenotypic genotypic ratio, this cross proves that characters are controlled by pair of alleles. These are the major points for what? Law of dominance, which is given by Mendel. What are the key features of law of dominance? Characters are controlled by pair of alleles. Okay, characters are controlled by pair of alleles, where one is dominant and the other will be recessive. In heterozygous condition, only dominant allele will be expressed here. Only dominant allele is expressed in heterozygous condition. Next, define and design a test cross. Okay, define and design a test cross. This has appeared in 2022 and 2016 as well. So here the test cross is what? Crossing of F1 hybrid. Okay, crossing of F1 hybrid generation, which is heterozygous individual with a homozygous recessive parent. This is known as test cross. What is back cross? Crossing of F1 generation, which is heterozygous dominant trait, with any of the parental generation is known as back cross. Fine, right? Back cross and test cross you have to be clear with. And here many a times the questions will be asked why the test cross should be conducted. You should know the clear reason why test cross. Okay, to identify the genotypic ratio of the F1 generation, test cross is conducted to identify the genotype of F1 generation. So here if you see in F1 generation, it helps us to find out whether the dominant phenotype is homozygous dominant or heterozygous dominant, and test cross is special type of back cross. Test cross is also known as special type of back cross. If the progeny shows both dominant and recessive phenotypes in equal proportion, the individual will be heterozygous. Okay. If all offspring show only dominant trait, then the tested individual is known as homozygous dominant. Okay, all the individuals, all the offspring show only dominant trait, then it is known as homozygous individual. If both dominant and recessive condition is observed, then it is heterozygous. Here we go. Okay, here we go with two phenotypes of F1 generation.
You read.
Let us suppose in F1 generation, we have two conditions. Heterozygous condition,
The walls of the gene organs
both
are
of
state of tonus.
recessive parent, small t, small t, small t. With recessive parents, it has crossed. Here, if you see the
extended to 7, 9 years. The organ
it will be
having
capital T, small t, capital T,
glands, liver, bladder,
or
pancreas.
homozygous dominant only. So here the phenotypic
That will be in the absorption.
will be tall phenotype only. But here if you see, when heterozygous individual, heterozygous F1 individual is crossed with homozygous recessive parent, when it is crossed with homozygous recessive parent, here you will get capital T, small t, small t, small t. Okay, so you will get 1:1 ratio. Okay, 1:1 ratio.
50 mg.
Completely tall, one completely heterozygous tall is to homozygous dwarf. Heterozygous tall is to homozygous dwarf condition. Okay, which is completely 1:1 ratio. Clear? Fine. And then this is the genotypic and phenotypic ratio. A test cross is the cross between F1 hybrid and recessive parent. This is to determine the genotype of the hybrid. This gives phenotypic ratio, that is 1:1 when the hybrid is heterozygous. I hope you are clear with law of dominance and test cross, right? Now, third point. For each question I am giving you a proper evidence, like what are the keynotes that you have to observe for your MCET and NEET as well. The preparation should go parallel, students. Never it should be a preparation separately for IPE where you are mugging up the questions. Never it should be like that. Okay. So third, if you see, mention the advantages of selecting pea plant for experiment by Mendel. Why Mendel has chosen pea plant? Okay, that is the question. Yes, here we go. What are the reasons for choosing Pisum sativum as the model organism for the conducting of experiments by Mendel? Fine. First is distinct contrasting characters. The characters are very evident, okay, very clear. Pea plant will show clear variations like tall and dwarf, round and wrinkled sheets, yellow and green cotyledons. As such, 7 contrasting traits were picked up. Each variant is clearly predictable, okay, dominant phenotype and recessive phenotype is clearly predictable. So it is the first foremost reason is what? Distinct contrasting characters. Second is annual plants with short life cycle. Okay, pea plant, it will not take years together for growing. Okay, it is an annual plant which has a very short life cycle. It completes its life cycle in just few months, allowing Mendel to observe several generations quickly, like F1 generation and F2 generation. And third, it is easy to cultivate. This grows in the garden. Right. It's easy to cultivate. This easily grows in the garden soil. Next is
pieces from the anus.
Productive flowers. Like each flower will have both male reproductive structures and female reproductive structures. Each flower has both stamens and pistil and both self-fertilization and easy cross-fertilization. Next is about large and conspicuous flowers are present in the pea plant. Large and conspicuous flowers. And next reason is self-pollination for pure lines. Many at times crossing with same plant for same trait. Then it will give pure lines like pure homozygous tall and pure homozygous dwarf. For one character, pure lines are observed. Self-pollination will ensure the purity. Next is easy cross-pollination. By just removing the anthers by the process of emasculation, by removing anthers from a bisexual flower, it is easy to conduct the cross-pollination. Next, this produces many seeds. Many seeds are produced. Each pod gives several seeds, providing large sample size for accurate statistical study. This gives you an accurate statistical study. Next, explain incomplete dominance with an example. Okay, incomplete dominant with the example. Okay, this has appeared in 2024, 23, 22, 17, 18, 19. Okay, multiple years the questions are asked. Like here you see incomplete dominance is the condition in which neither allele is completely dominant. No allele is completely dominant and no allele is completely recessive. This results in an intermediate phenotype in the heterozygote. This results in an intermediate phenotype. And here in incomplete dominance, both the genotypic ratio and phenotypic ratio will be one same, which is 1:2:1. Okay. So what is the model organism which is to prove the incomplete dominance? Flower color from snapdragon or anther matches or 4 o'clock plant, that is Mirabilis gelapa, cross between red color flowers and white color flowers which are homozygous dominant and homozygous recessive will give you the F generation.
You all has to remember, you know,
smaller, which is heterozygous dominant. Here if you see the parental generation, true breeding is crossed with true breeding recessive. True breeding dominant trait is crossed with true breeding recessive trait, which produces pink flowers in F1 generation. Followed by, here you see F1 generation, all plants are heterozygous dominant, showing intermediate pink color.
Blood supply happens.
Actual expression of
Blood supply happens. So in
F2
words you can
or selfing of F1 generation. Okay, F2 generation is produced out by selfing of F1 generation. That is heterozygous dominant parents. Both are heterozygous dominant parents. That will result in
the structure wise, it is just because
capital capital small r and small r small r. As such, 3 different genotypes are produced.
For structural
okay. Here in incomplete dominance, dominant allele is not completely masking the effect of the recessive allele. So here you will get one homozygous dominant and heterozygous dominant too. Both are showing pink flowers only. Dominant allele is not completely masking the effect of the recessive allele here. That is the whole concept. And two recessive alleles will give you recessive phenotype also, that is white flowers. So here both the genotypic and phenotypic ratio is one and the same in incomplete dominance. Many a times in your MSET and NEET also the questions are put on incomplete dominance and codominance as well. Fine, right? So here define law of segregation and law of independent assortment. Whenever concepts or two questions are
called as muscularis mucosa,
like either asking about the comparative analysis or asking about the differences between two domains, then you have to be clear with the table. Okay, make sure you are providing a tabulated form of answer whenever you are delivering a comparative analysis or differences between two concepts. Make sure your answer will be in the tabulated form. First is of segregation, second is law of independent assortment. Law of segregation and law of independent assortment. Two alleles of genes, they will separate from each other during
the nervous system.
They will separate from each other during gamete formation, so that each gamete will give only one allele of a pair. Okay. In independent assortment, the segregation of one pair of alleles occurs independently of segregation of another pair of alleles during gamete formation. Law of segregation is the first law, and this law is known as law of purity of gametes, and law of segregation is universal without any exceptions, whereas law of independent assortment is the second Mendel's law. This law of segregation is proposed from monohybrid cross, whereas law of independent assortment is proposed from dihybrid cross. Fine, right? And then here in a heterozygous plant, gametes are formed which are capital T and small t. In dihybrid plants, the alleles or gametes which are formed are these. Fine, right? Next, these are two alleles from the monohybrid cross. Sorry, these are the two gametes that are from monohybrid cross. These will be the gametes of dihybrid cross. Law of segregation will ensure pure alleles are transmitted offsprings without any blending. There is no mix or blend of alleles which are present in a factor. But here this ensures the independent inheritance of different traits in law of independent assortment. This deals with separation of alleles of a single gene, whereas this deals with independent behavior of alleles in two or more genes. And this is seen in all sexually reproducing organisms, whereas law of independent assortment is observed in organism which is showing dihybrid or polyhybrid inheritance.
We call this.
Read out anywhere and then
part. Right. So
see, I think the question is about differentiation. Differentiate between the following: dominant recessive traits, homozygous heterozygous condition, monohybrid and dihybrid cross. Look at the differences. Difference between dominant and recessive alleles. Dominant allele, here this is known as unmodified allele. In your NCERT, modified allele is the term which is used for the recessive alleles. So here you see the unmodified allele of a gene that expresses its trait even in the presence of contrasting allele. But here in the presence of contrasting allele, the effect of the recessive allele is masked.
You observe
modified allele gene, this fails to express its trait in the presence of dominant allele. Very clear, like you can easily make out what is dominant allele and recessive allele, but when you are presenting it in the form of answer, terminology is very important. This is expressed both in,
you can say, small funnel shaped structure.
Dominant allele. Recessive allele is expressed only in
below the tooth.
The dominant
generally call it as a frenulum.
The recessive trait, this remains unaffected during F1 generation and this reappears during the F2 generation. And then
and lip, that is the gum. What is that?
whereas in pea plant, dwarf, that is small t, this will be recessive. Clear till here? Then if you see difference between homozygous and heterozygous condition. Homozygous condition, this is the condition in which both alleles of a gene are identical to each other. Here in heterozygous condition, the alleles are identical to each other or they are different. Okay, and here these are represented as such, and this will be represented in the heterozygous condition. This produces out two gametes. Homozygous condition only produces single gamete. Okay, and here this is completely pure tall and pure dwarf is seen. Their hybrid tall is also present in the heterozygous condition. Differentiate between monohybrid and dihybrid. Monohybrid is nothing but one gene inheritance. Dihybrid we will say it as two gene inheritance. Okay, monohybrid is one gene inheritance where this involves just one pair of contrasting traits, whereas dihybrid, here is the cross where two pairs of contrasting traits are present. Here organism is heterozygous in just one locus, organism is heterozygous in two loci. This cross is in between dominant alleles, dominant and recessive allele, just in between two alleles. But this is the cross in between both the traits, that is seed color and seed shape. And then you see this produces two types of gametes. Here it produces four types of gametes. One is red, yellow seed color and round seed shape, yellow and round seed. Second type of gamete is yellow seed with wrinkled shape of seed and green seed with round seed shape, wrinkled seed with green seed, wrinkled seed shape and green seed color. These are the four gametes which are being taken in the dihybrid cross. Fine, right? Yeah. And then you see, explain the codominance with examples.
Comes out, food also spills out.
Dominance,
The reason is what?
which we
got disturbed. Whenever we are eating normally without having any conversation, food generally passes, it never.
remember
It is just because
incomplete dominance. These two
What is the
major concepts for you to NEET as well. So when you are exercising with this question, parallelly exercise the objective regarding the incomplete dominance and codominance. Here if you see, the question has appeared multiple years in 2023, 2020 and 2016. Answer you go through. Codominance, this is the phenomenon in which both alleles of a gene, they will express themselves equally. They express themselves equally in a
between
heterozygote. Neither allele is completely dominant nor they are recessive. Fine. In codominance, if you see, the heterozygote, this exhibits both parental traits simultaneously. Both the parental traits are expressed simultaneously without blending or mixing. Fine.
Right.
Blood grouping in humans where two alleles are observed, that is IA, IB genotype.
Mouth.
Show AB blood group. So in the AB blood group, when two alleles are
That's the book.
both the alleles are equally expressing, okay, that they are equally expressing, then it is addressed as what? AB blood grouping is for codominant.
Two palate.
Fine, right. And next,
Palate.
coat.
Palate is
Both A and B antigens are expressed.
bonds. Right. Then soft palate, it is exclusively made up of muscular.
Fine. Next, seed coat pattern in the lentils, that is lens culinaris. When a spotted plant is crossed with a dotted plant, in the F1 generation, you can see the seeds both with spotted and dotted condition. Okay, seed patterns will show both the conditions, that is spot and dot. Even if you are not remembering the second example, first example you make sure that you are doing this, because this is the standard example for this codominance. Fine, right? Which is in text as well. See about AB blood grouping. AB blood group will have both the antigens A and B. Next, here, write a brief note on chromosomal mutations and gene mutations. Chromosomal mutations and
second fluid. We call serum. There are
sudden heritable changes that occur due to
glands
of chromosome number or structures. alteration of chromosome number or structure, that is chromosomal
Will
mutation.
create
This is first
immune
discovered by Hugo de Vries in the plant Oenothera. Whereas gene mutations, these are sudden heritable changes. They will occur in DNA base sequences within a gene. This is also studied as a part of Hugo de Vries mutation theory. This is also studied as a part of Hugo de Vries mutation theory. Here, this affects whole chromosomes or large segment of chromosomes. Whole or large segments of chromosomes is affected. Gene mutations, this affects individual genes or few pairs of the genes. Here we go with this change in chromosome number. You can see as aneuploidy.
These three glands.
Okay, aneuploidy and polyploidy. Change in chromosome structure you see. Deletion, duplication,
All information
and translocation. This is the change in chromosome structure. Here you get mutations. Point mutation is a mutation where a change in single base pair. Frame shift mutation, this is because of the insertion and deletion of one or more base pairs. What are these diseases that occur? Genetic disorders or chromosomal disorders that occur due to chromosomal mutations. Includes Down syndrome. This is because of trisomy of chromosome 21, and Turner syndrome, which is because of monosomy. Fine. Next, gene mutations. Here you can see sickle cell anemia. This is due to substitution of one of the base pair. And chromosomal mutation. This affects many genes at once because entire chromosomal segments are altered over here. But here this affects only one specific gene or a small DNA segment. This causes large scale changes in the phenotype or abnormalities, whereas this causes small scale genetic changes, some leading to some defective protein. Fine, right? And then next chapter is from molecular basis of inheritance. Nucleosome model is very major concept both for your objective and your subjective exam. See, nucleosome model many a times asked in 24, 23, 23. Even in long answer type of questions, nucleosome model is expected. So here if you see nucleosome model, here you have to remember about few things. A single nucleosome model has how many histone proteins? Okay, single model or a single nucleosome also having how many histone proteins? Histone octamer, DNA which is negatively charged is completely winding up to the positively charged histone octamers. Here histone octamer, single histone octamer will be containing two sets of 4 proteins, that is H2A, H2B, H3 and H4. Two sets of these 4 proteins will make up one histone octamer. Okay, one histone octamer. Here the negatively charged DNA is wrapped in the positively charged histone proteins, and this binds up 2 nucleosome models. The linker DNA will connect up 2 nucleosomes. Linker DNA is H1 histone protein, that is H1 histone protein. Fine, right? Clear with this? Next. Nucleosomes, these are the fundamental and structural units of chromatin. They are found in nucleus
for water.
Histone proteins, they will organize to form a core of how many molecules?
Right. Now you all has to
form 8 molecules which are known as histone octamer. They are known as histone octamer. Histone proteins, they have positively charged amino acids like what? Like
what?
Fine, right? So histone octamer, they have 2 molecules, 2 sets of molecules like H2A, H2B, H3 and H4 molecules. Negatively charged DNA is wrapped around positively charged histone octamer, which is a nucleosome. Each nucleosome will contain 200 base pairs of DNA helix. Each nucleosome contains 200 pairs of DNA helix. Here H1 histone protein, they will lie outside the core, and this will help to seal the DNA, which turns by binding. Fine, where DNA enters and exits the core. When you observe on an electron microscope, you can see the chromatin arrangement as beads on string structure. This is like beads on string structure.
Present in the saliva, which is immunoglobulin A.
Together to form chromatin structure.
See,
fine, right? Beads on string structure for the coils, and this gets condensed to form chromatin fibers, which finally holds into the chromosome during cell division. Here one thing is very important, like what are the histone proteins, what are the amino acids, which is giving the basic nature, and third is how many base pairs are present. Base pairs of DNA helix are
you can found
each nucleosome. This is very important. And then this is completely about the nucleosome model. What is
it is present in the external situation, whatever the pathogen.
Okay. Differences I'll let you know, ma.
That can
First about sugar. Here in DNA
point
deoxyribose sugar, where at the second carbon of
do not
sugar, OH group is absent. Whereas in ribose sugar, at the second carbon of the ribose sugar, which is pentose, OH group is
single.
So here the molecular formula is C5H10O5 in ribose.
Very good. It breaks.
The formula will be
where the bacterial cell
O4. Absence of oxygen group at the second carbon. Next,
down in this particular
the nitrogen bases DNA include
globulin A, lysozyme.
These are the purines. Find cytosine and
the tacton. Now you have
pyrimidines.
And very important.
Okay, two purines and pyrimidines are present. In RNA QC, RNA, where adenine and guanine are the purines which are present, whereas thymine, cytosine and uracil are the pyrimidines which are present in the RNA. These are the purines and pyrimidines in the DNA. These are the purines and pyrimidines in the RNA which are present. So here thymine in the place of uracil will give additional stability to the DNA, whereas uracil which is present in the RNA will make it reactive. Fine, right? And one free oxygen group which is present at the second carbon of ribose sugar will make it more reactive and mutable in the RNA. These are the not differences. And DNA is found nucleus. Some parts of it are found in mitochondria and chloroplast. As they are autonomous cell organelles, they have double-stranded circular DNA. Whereas here in the RNA, they are found mostly in the cytoplasm and small amounts of it are present in the nucleus. Clear? Then look at this question. What are the contributions of George Gamow, Hargobind Khorana, Marshall Nerdberg in deciphering the genetic code? Okay. At times statement based questions on this concept are asked in your NEET PYQs. You can check it out. So here, contributions in deciphering the genetic code. First with George Gamow. George Gamow has proposed genetic code. He has proposed the genetic code. This must be a triplet in nature, meaning
if the salivary enzyme amylase
code for one amino acid. 3 nucleotides together will code for one amino acid. He mathematically suggests a combination of 4 bases. The combination of 4 bases taken 3 at a time when 4 bases put together forms how many?
R
in codons. This could code for all the amino acids. 20 amino acids. This idea laid the foundation for understanding the triplet nature of the genetic code. This is given picture of the
cofactor for the amide. When we consider,
he has chemically
the major function buffer.
This is with specific and known sequences of the DNA. He used homopolymers, which is this base for.
Not conclude that it is completely acidic. It is
and copolymers, which is UUC and CCA. This is to determine the codons corresponding to which amino acids. His experiments provided definite proof to specific codon amino acid relationships. Fine. This confirms specificity and universality of the genetic code. Next is Marshall-Nienberg. Marshall-Nienberg developed a cell-free protein system that enables the
what cell are made up of?
observed protein formation and synthetic RNA templates. Fine. And then he discovered uRNA, polyuRNA. He directed the synthesis of phenylalanine, which proves that you codes for phenylalanine, the first codon ever deciphered. Next, using this enzyme, polynucleotide phosphorylase, and he and his colleagues synthesized RNA with defined sequences, leading to complete decoding of the genetic code. So first it is H.G. Khorana who has synthesized RNA molecules with specific bases. Next, copolymers are being synthesized. Marshall-Nienberg. Fine, right? First explain about George Gamow discoveries, followed by homopolymers and copolymers, which are given by H.G. Khorana and Marshall-Nienberg. Last question of today's session on the types of RNA polymerases, which have asked in multiple years like 2023 and 2017. Here we go. There are 3 types of RNA polymerases which are found in eukaryotic cells, and each polymerase is responsible for transcribing a single set of RNAs. Look at here. Types of RNA polymerases, we have 3 types. RNA polymerase, RNA polymerase 2 and RNA polymerase 3. What is their major functions and what are the products synthesized from them is important. RNA polymerase 1, which transcribes genes in the nucleolus. It is better if you write the location also in the nucleolus. Inside the nucleus, in the nucleolus, RNA is being synthesized. This produces 28S, 18S and 5.8
membrane, so it always moist.
This will produce 28S, 18S and 5.8S.
Then what happens?
All the components of the ribosome. Next, RNA
you must observe the people who are having slurred speech, right? Those people also,
this transcribes structural and protein coding genes. Transcribes structural and protein coding genes. This produces precursor of mRNA or hnRNA, which later forms vector mRNAs, which also synthesizes some small nuclear RNAs, which is known as snRNAs, small nuclear RNAs. And then you see RNA polymerase 3. This transcribes small RNA. They will transcribe RNA genes. So RNA polymerase 3
by the synthesis of stimulation
is also known as soluble RNA. This produces tRNA, 5S RNA and snRNAs, which play major role in splicing. Right. And 5S rRNA, they are one of the proteins present on the ribosome. Fine. So these are the products that are synthesized, main function and the location where are they being synthesized or produced. So both tRNA and mRNA, that is messenger RNA, are synthesized inside the nucleoplasm. They are synthesized in nucleoplasm. So with this, the complete session on principles of inheritance and molecular basis of inheritance is done. So these are the short answer type of questions which are to be focused, along with what concepts we have done: nucleosome model, types of RNA polymerases, the scientists who have deciphered the genetic code from molecular basis of inheritance, codominance, incomplete dominance, loss of inheritance like the law of dominance, segregation, independent assortment. These are the concepts which are highlighted from principles of inheritance. Okay, the concepts are important for your MCET and NEET as well. Now when you are going through these concepts for your IPE, practice the objective on the same note, okay, which will make your preparation on the parallel tracks. So with this, the complete info on the genetics is done. We'll see you in the next class.